All right, good morning, everyone. Thanks to Luke and for inviting me for this presentation. I'm going to talk about a little bit of the results of some trials we did across Illinois. Some of them were even on this research station back over there for a couple of years on how to manage soybean seedling diseases. But I'm going to also talk about the corn seedling diseases. But first, my name is Santiago Mideros. I'm an assistant professor of plant pathology at the University of Illinois in the Department of Crop Sciences. And a little bit of a reminder, perhaps, of what these seedling diseases look like, just the symptoms, or how do we identify them. The main thing is poor emergence that is going to lead to poor stands later on the season. There's usually some patches of damage. Then we have a lot of, it could also shown as a smaller, on the smaller regions, right, or on smaller areas of your crop, as shown as the skips or yellowish plants or stunted seedlings that even can have as an individual, it could be at an individual place, right? So you just have one missing seed in some places. You could also notice some uneven plant height, and you can see that in crops that are a little bit more advanced than what's on the picture. But if you start seeing like waviness, in many cases it could be because they are being the roots have been infected and it's slowing down the growth in certain areas of those for those fields. There's also wilting, or leaf necrosis and scorching, and this is a very sad-looking soybean plant, usually wilting when it's not wilting that is not due to issues of lack of water, right? Now you might be thinking I'm showing all soybean pictures here, but all the titles there came straight from the compendium of corn diseases. So those are things that Don White and Gary Monfolk have written about exactly how the seedling diseases happen on corn, right? And I'm showing you here pictures of soybeans because they look the same. And also I'm showing you pictures of soybeans because my research was on soybeans thanks to the support of the North Central Research Program that gave us some money to test that. But I'll be talking about all that I'm talking here applies to both. And I'll talk a bit more on why is that. The other symptom that we get for this disease is damping off. It could be pre-emergence or post-emergence. With pre-emergence damping off, you get seedlings or seedlings that do not manage to come through the soil, obviously. In many cases, they produce these dark patches on the seed, and that is even if you can find the seed. In many cases, you cannot find it because it has melted into the soil, right? Because they get rotten. And post-emergence is when it just comes out of the soil and then it just falls down because of some sort of infection. In some cases, if it is very wet, you can even see some mold growing around them. And some people that are with a lot of training, you can even start differentiating a few of the different pathogens that can cause those issues. So there's many organisms that cause these diseases, right? On corn, we have about a half dozen species of fusarium that can be causing these. There's also the ear rot pathogens that can be infecting the seeds, Penicillium, Aspergillus, Rhizopus. A few species of each one of those can cause problems. Pythium is one of the big ones, right? Pythium, there is about 3 dozen species of Pythium that cause seedling diseases. So there's a large variety of those. And there's a few isolates of a few species of Rhizoctonia that can also cause disease on corn. For soybean, pretty much the same, except for soybean we have to add a few other two that are very, especially Phytophthora sorgi, very important plant pathogen. The Phytophthora also somewhat related to Pythium. These are water molds. They really like wet conditions. And a big issue with soybeans and corn. And also Rhizoctonia, right? So we have almost all of the most important pathogens can cause disease on both corn and soybeans. So what do we do about them? I recommend always to look into an integrated pest management system. For that, I'm just putting here the four first steps of IPM practice. Identify them. I recommend you can identify them based on symptoms. Just put the pictures there on your handouts also, and that's how you can see if there is an issue with seedling diseases. Don't confuse them with water damage, right? Of course, if the field or areas of the field are underwater for a couple weeks, yeah, there's going to be issues, right? And it's not necessarily the seedling diseases. But if the water the flooding is not that major and you have some infection and the symptoms, then you're dealing with a disease. The next step is some sampling, right? If you're implementing IPM, you need to do a little bit more of work on that. You need to clearly determine what is the distribution of the problem, identify which areas of the fields are problematic or which fields are problematic, and then you can do a lot more as far as management if you have that type of information. It's important to dig the plants carefully to look for those rotted seeds. If we're looking at the rotted seeds, we can start identifying the pathogens there and have more data from them. You can collect the whole plants. You could do more identification yourselves, or you can send them to the plant clinic or to myself. We are all the time getting samples and trying to characterize more carefully which are the species that are causing disease here in Illinois. The next step, of course, is the decision making, and obviously with IPM, what we want to do is to find a way to manage our disease that has the lowest possible cost. It is a bit complicated with seedling diseases because there's so many pathogens out there, so many issues that could be causing the problem. And this it's going to depend in a certain way for every farming operation should develop a system on how to make these decisions, right? What applies to a small farm operation is different to what applies to a larger farming operation, right? So we have to make a system that is consistent year over year, because that way we can improve year over year, right, on what are we going to do for the management. And I have in a couple more slides a list of the things that are recommended, right, for these that you could decide from. And last but not least, and probably the most complicated, is the economic injury levels. I was going to say first, and on your handouts it says not available for seedling diseases. It's not completely true, but in part yes, because there is some break-even analysis that have been done, especially for soybean seed treatments. But the results of those break-even analyses are complicated, right? In many cases, what it comes out is that it depends on your variety, right? So that's not quite enough for an economic injury level. What I would like to see is something like, okay, so when we see a percentage of damage on a field, that is it's enough for to recommend a replanting of that area, or is enough to recommend a replanting of the complete field, etc. Right? So that will be a more a specific economic injury level application. But people are working on those, except there is not, as far as I know, a complete economic injury level study into these seedling diseases. Right? So on the sampling area, that second spot, I wanted to also share some of the results we have found from these samples that people have been sending to us, or we've collected from fields across Illinois. And this is again is for the soybean samples that we've been getting from both soils and diseased plants. And what we find the most in Illinois is Pythium ultimum, one of those Pythium water molds that are causing the seedling diseases on soybeans, right? Followed by Phytophthora, and then a few other Pythium species. Those are the more common, right? To do this, we do microscopy, we do DNA analysis to have a clear idea of what is exactly the species causing the problem. Now, on that next third step of how we decide what to do, these are the recommended practices. Of course, for both corn and soybean, we have to start with high-quality seed. Then for soybean, we have an extra tool, which is there is some RPS genes, there are some resistance genes but they work only against Phytophthora soji, right? If it is Pythium, if it is Fusarium, is anything else, they are not going to work out very well. But given that Phytophthora is one of the top issues with soybeans, then it offers a level of protection, right? If that is the issue on your field. The next recommended thing is seed treatments, and I have some data also to show they work very well, especially if we have several active ingredients. There's also an issue about the temperature. Planting corn above 55 Fahrenheit on soil temperature helps significantly if you're having issues with seedling diseases. The same goes for soybeans. It's not as specific, but people know to avoid wet and cold conditions for soybean planting, because they favor those wet and cold conditions favor a lot of Pythium species that can cause the disease. And last, but probably the most expensive also, is to improve drainage. If a field is having a lot of drainage issues, you're going to have more seedling diseases, right? I was saying that for Phytophthora, we have the advantage of having some RPS genes available, but not all the RPS genes are the same, unfortunately. These RPS genes for controlling Phytophthora soji are very specific to certain isolates of Phytophthora, right? So with those same samples that we've been collecting from Illinois, we've been testing them and see if they are able to overcome which RPS genes, right? And this is what I'm showing here, just the percentage of isolates collected in the state that are able to cause disease. And every single one of them, for example, is able to overcome the RPS1B gene, right? So don't use that one for sure. But there's some very effective. On the other hand, RPS3B, 4, 6 are pretty good. Others that are very common, such as 1K, it seems like the efficacy of that gene, it's falling down. It's less than 50% these days with the current samples, right? So if you have the RPS 1K gene, the chances that it's going to work for you are less than 50%. So for seed treatments, again, I said they work very well. There is several active ingredients that are put on seed treatments these days for both soybeans and corn. Metalaxyl, mefenoxam, or ethaboxam, they are specific for Pythium and Phytophthora. Okay. Other active ingredients that you see are iconazole or thiabendazole. They target the true fungi, such as Fusarium, Rhizoctonia, and many others that were on the list of the causal agents back there. There is one that hits a little bit of both, azocystrobin or the strobilurins. They hit both Pythium and Phytophthora, but also fungi. Seed treatments these days also come with insecticides, nematicides, and some even with biologicals. I could give a talk about each one of those. Actually, maybe not me, because I'm not an expert very much on insects, but people could give a whole talk about how to use those insects on seed treatments or not. But the important thing that I will recommend is that there has to be more than one active ingredient on your seed treatments. And what I would recommend is one from the first stop, from the first group, something that targets Pythium and Phytophthora, and something that targets the other fungi, right? The first group, so metalaxyl, mefenoxam, or ethaboxam, either of those, plus something that hits Fusarium, such as iconazole or thiabendazole. Why is that? Well, not all, just like for the RPS genes, are not all effective against all the strains of Phytophthora. The seed treatments, each one of those fungicide active ingredients are not effective against everything that is out there. For example, also from the samples that we've been collecting on Pythium, we tested them against different fungicides, in this case azocystrobin here. And what this shows is just the frequency of the isolates and at which concentration of the fungicide they are controlled, right? So how much of the fungicide, how much of the product you need to control that specific isolate. So for example, here I'm just showing that one isolate of Pythium ultimum, it requires a very low dose, almost close, it's not zero, but it's just because I'm contracting the figure here, it's showing a zero. A very close, very low dose of azocystrobin is enough to stop that Pythium ultimum. And another strain just needs a little bit more. But then, yeah, azocystrobin very good at controlling then many isolates of Pythium aphanidermatum, many isolates of Pythium disodicum, good with Pythium ultimum. You need a little bit more to control Pythium irregulare, but when it comes to Pythium vexans, for example, one of the isolates we find a lot from Illinois soils, not very good, right? It's not effective. Etavoxam the same, but yeah, it kind of switches, right? The species in this case. Pythium vexans that was not very good with azocystrobin. That was not being controlled with azocystrobin. It gets very well controlled with etavoxam, but Pythium aphanidermatum, it's like you're giving it water. Doesn't help at all. So yeah, changes. Therefore, important to have seed treatments with different active ingredients. So we did some, as I was saying, we did some trials, multi-locations. Here in Ore was one of those locations about using these seed treatments and different varieties of soybean varieties with different RPS genes to test how they were going to work. And yes, they indeed, the seed treatment that we had that had etavoxam, metalaxyl, and iconazole was very effective. It gave us significant levels of control for plant stands in the early season and also on the late season. Even yield responses, there were some increases on, yield protection, I would say, on those environments that had high disease pressure, so environments that received a lot of rain early on the season. We got significant yield protection for the seed treatment. The RPS genes worked on some fields, but not in others, right? And that makes sense because it depends which pathogen and which race of the pathogen you have, whether the RPS gene is going to work or not, okay? Because they are race specific, right? So they did work in some cases, but also partial resistance on soybean against Phytophthora was pretty effective at controlling at a low level the issues with seedling diseases. Okay, so I've talked enough. Are there any questions that I could answer?